confirms if anybody's thinking about asking. Today's session is being recorded and the link to the recording will be sent round shortly after the session today. So if you have to leave early for any reason, or if you have any connection problems or whatever, or if there's colleagues who would like to see it, then you'll have the link later to be able to have a look at the presentation in your own time. So, welcome to the ScHARR mini masterclass. I'm already recognising names now. Every time we run one of these, the number of names in the names in the participant list are becoming familiar, or at least some of them are. So it's nice that a few of you are kind of joining us from month to month. Just a reminder, if you're interested in our Masters in Public Health diploma or certificate program in public health, there's a link on the screen. I'll paste that link in the chat in a moment as well if anybody's interested in following it up. Stephen, who's our speaker today, teaches on that as well as other programs that we offer in ScHARR. All of the programs that we offer in ScHARR are postgraduate programs, so we have master's students and postgraduate research students, PhD students. Also, before we start, just a reminder about February's mini masterclass. This is going to be with Dr. Liz Croot. She's a senior research fellow and the director of learning and teaching at ScHARR. She's going to be joined by Jodie Bradley and Vicky Farnsworth with a webinar about doing research with people with learning disabilities. So again, the link's on the screen, and I'll put it in the chat box in a moment once Stephen started his presentation. So if you want to book ahead for February, please feel free to do so. But today's webinar is by Professor Stephen Walters. I've known Stephen the whole time I've been at ScHARR, and I've been at ScHARR for nearly 18 years now. But Stephen beat me to it. He joined ScHARR in January 1995, so that's the year after ScHARR was founded, and became a professor in 2009. He was also the first director of the Design, Trials and Statistics section in ScHARR from 2012 to 2018. He's a chartered statistician with over 25 years of experience in health services research and technology assessment. And the main focus of his research is in the design, conduct, analysis and reporting of trials of complex interventions, and the design, assessment, analysis and interpretation of patient-reported outcome measures in clinical trials. He's been a co-investigator for more than 20 RCTs of non-pharmaceutical interventions and has co-authored over 300 publications, including 200 in refereed journals and 4 books. And he teaches across ScHARR's range of postgraduate programs and has been the recipient of the annual ScHARR Teaching Award, which is nominated by students. He's currently an NIHR senior investigator and was also a member of the NIHR Health Technology Assessment, Clinical Evaluation and Trials Board from 2011 to 2017. And today he's here to talk about the ABC of RCTs. So it's my pleasure to hand over to Stephen. I'm just going to switch over the slides to Stephen's presentation. Okay, Stephen, over to you. Oh, and sorry, I should say before Stephen starts, I do apologise, Stephen. If you have questions, we're going to take the questions at the end, but it's fine to post questions in the chat box at any time during the presentation, and then I'll come on at the end and I'll pick through some of the questions and we'll go through them. Because sometimes people like to comment as the session goes on, we'd like you to just put Q in front of your question in the chat box if you're asking a question that you'd like Stephen to respond to. If you just want to make a comment, obviously no need to do that. But if you're putting a question in, just put Q in front of the question so we know that it's a question. Okay, over to you, Stephen.
Thank you for that introduction. Thank you, yes. So this talk is going to be about the ABC of RCTs. So what we're going to talk about then really is the aims. Is, as I say, I needed a catchy title and I couldn't think of anything, so I thought I'd talk about the ABC of RCTs. And there's lots of news about randomized controlled trials now. Seem to be what's going on now in health services research and evaluation. And thankfully, most of the new trials that are being conducted are following these principles. So what am I going to talk about? Well, I'm going to talk about, try and emphasize very briefly the importance of randomized controlled trials in evaluating alternative treatments or interventions. I'm going to talk briefly about the rationale for randomizing and give you some links and videos that you can look at if you want to really know the different methods of randomizing patients. I'm going to mainly talk about the ABC of randomized controlled trials. That's allocate at random, blind assessment of the outcomes, and having a control group, a contemporaneous control group. I'm briefly going to mention about the trial protocol and talk about different types of design when you have trials that are basically wanting to establish whether a new treatment is better than another, from trials that have a slightly different aim or objective, which is to seek equivalence or non-inferiority. And if I've got time, we'll briefly talk about some different types of randomized controlled trials, such as factorial, cluster, and crossover. And I'll maybe give you some links to also the sort of checklist if you're designing a trial, analysing, or even the reports of a trial that you can look at as well. So that's the plan. So the key idea really in a clinical trial and a trial is really to compare groups of patients who really should only differ with respect to their treatment. And really, you really want to keep everything the same, if at all possible, except for the treatment they receive. Because if the groups differ in some other way, then the comparison of those treatments may be biased. And as I've already alluded to, the two treatments should be investigated concurrently. Allocation of treatments to patients should be a random process, and neither the patient nor the clinician should know which treatment was received. So that actually is the CAB randomized controlled trials, contemporaneous control group, allocation at random, and blind assessment of outcome. But I thought ABC sounded a little bit better. And what we call this so-called double-blind randomized controlled trial is typically taken as sort of the gold standard against which we should try and judge the quality of a randomized controlled trial. So when we undertake randomization basically is just a method of by chance of allocating patients to the different treatments to ensure that each patient has a sort of a known chance. I mean, this is usually equal, but not necessarily always the case, of being allocated to the available treatments that you've got. And it should also ensure that the treatment you receive cannot be predicted. And this, I'll explain a bit later on, will hopefully prevent issues of bias and confounding. Now, the crucial thing lots of people tend to forget, and even I tend to forget that, is just because you randomize participants to alternative treatments, you actually also need to randomize them, but make sure you conceal. And so up to the point of consent and actually getting the treatment, the person that's recruiting the patient doesn't actually know which treatment they're going to get. So you have to sort of make sure you also conceal the randomization sequence. So don't leave it laying around, or use, in most modern trials now, use a computer-based, internet-based randomization service. Right. So I could spend the whole of the talk talking about the history of randomized controlled trials, but I suppose that the first sort of what we would call proper randomized controlled trial that would be widely recognized as a randomized controlled trial actually happened relatively recently. I suppose 73 years ago is relatively recent. It was a trial in the United Kingdom funded by the Medical Research Council of streptomycin treatment of TB, tuberculosis, and this was published back in 1948. And why this is sort of widely recognized as the first sort of proper randomized controlled trial, and there have been lots of other trials before that perhaps had elements of randomization and blind assessment of outcome, but not both together, because this trial basically included concealed randomization and blind assessment of the outcome. And patients were not told before the admission to the hospital that they were actually going to get a special treatment, this new drug, streptomycin, and actually control patients didn't even know they were control patients sort of in a special study. And so I think the ethical issues have slightly moved on now, sort of 70 years later. I don't think you could get away with that. But it turns out there were 107 patients randomized, 5 got the new drug, and 52 got the control. And what it says in the methods of the paper is that the randomization was performed, I'll quote it here, by reference to a statistical series based on random sampling numbers drawn up for each sex at each center by Professor Bradford Hill. He was a professor of medical statistics. And the details of the series were unknown to any of the investigators and were contained in a set of sealed envelopes, each bearing on the outside only the name of the hospital and a number. So here we've got concealed randomization with the lists prepared in advance. So again, concealed allocation. And actually, the nature of the treatment meant you couldn't actually stop people knowing that they were getting bed rest or obviously getting the drug streptomycin. But actually, in terms of the assessment of the outcome, they had x-rays taken of the participants' lungs. These were then sort of examined independently or viewed by 2 radiologists and a clinician, each looking at the films independently and actually not knowing when they were viewing the outcomes, the x-ray of the patient's lungs, whether they were actually a streptomycin or a control case. And in this case, this is what we call blind assessment of the outcome. So that was widely regarded as one of the first sort of proper randomized controlled trials. Now, the crucial thing, if you're trying to design a study, particularly an evaluative study, where you want to compare 2 treatments or 2 interventions, then you basically need a good design that leads to internally and externally sort of valid comparisons and avoids what some people have referred to as the 3 enemies of the truth: chance, bias, and confounding. And really, if you're going to make want to evaluate a new treatment or technology, you really need some form of control group to make that comparison. Now, if you're designing a trial, well, actually, you need to write a sort of protocol, not necessarily a book by Frederick Forsyth, but actually the protocol is effectively a formal document specifying how the trial is to be conducted. And this protocol does provide sort of a reference for investigators when they're entering patients into a trial, and equally conducting the trial and then actually wanting to analyze the outcomes of the trial at the end, and perhaps even maybe stuff about how they're going to report the results as well. So the protocol is an important document that you should have if you're going to design a trial. Now, I suppose the key features of that protocol, and I suppose so the 3 fundamental aspects of that protocol and for a randomized controlled trial, which really is for all trials, should cover this. First of all, you have to decide what are eligible. So you need to think carefully about your inclusion and exclusion criteria. You also need to clearly define what the treatments are that you're going to evaluate and what the control treatment or usual care is. And equally, a third important point that you need to think about is how each patient's response or outcome to that treatment and intervention is going to be assessed. So really, there are 3 sort of key features of a protocol that you should really define: the eligible patients, what treatments you're going to evaluate, and how you're going to assess the patient's response to that treatment. And it turns out, sort of recently, there's actually been what's called the SPIRIT statement, standard protocol items, recommendations for interventional trials. And you've got the sort of the address there, the URL there, which tells you about the SPIRIT statement. But that's basically a checklist and a guidance of what items basically to include in a trial protocol, particularly for if you're having what we call an interventional trial, you're trying out a new treatment or a new technology. So that's there. I suppose with trials now, and even with this talk, it's always good to try and have a good acronym, as it were, for your guidelines or recommendations. So SPIRIT, standard protocol items, recommendations for interventional trials. So I'd recommend, if you're interested about sort of trials and protocols, to have a look at this guidelines and the checklist for that. So what I'll talk about for the next few slides is basically, we'll talk about the idea of the A for random allocation in randomized controlled trials, what you should do. And yeah, photograph here just to illustrate, well, actually the randomization process can be fairly simple. It could even be shuffle a pack of cards and select a card. You have some colored counters or beads, one colored one color, another colored another color, or even you have a dice. That's the chance element for the randomization that you use. But most randomization sequences now actually do use a computer package to do that. I have been involved in the past in a trial where actually there are only sort of 5 units to randomize, and I just do that, just simply toss a coin to decide on that. So it could be even as simple as that. So the crucial thing then is, I've talked about, well, randomized control trials, and the double-blind randomized control trial is widely recognized as the gold standard. But why do we need to randomize? Well, randomization, as I say, is a procedure in which the assignment of a subject or patient to the different treatments is decided by chance, so that basically the person that's recruiting that participant and ultimately treating that patient basically cannot predict in advance what treatment they're going to be given. And I know Mike Campbell and myself say this in our sort of textbook that we've written, that we probably think it's one of the most important innovations that statisticians have given to medical science. It was sort of introduced actually by R.A. Fisher in terms of agricultural experiments, but as we've just seen with the MRC streptomycin trial, it was really Austin Bradford Hill that developed it and got it into use for trials on patients and human subjects. And as I've said, randomization and chance could mean the toss of a coin, the draw of a card, or in the ideal world now we tend to use a computer-generated random number. The crucial point is that the investigator does not influence who gets which of the alternative treatments. And the main point of this really is that lots of experience has shown that if you have any other method, then investigators tend to either consciously or subconsciously, they tend to there's a potential bias in the allocation of treatments. If they're recruiting the patients or treating the patients, they tend to make decisions if they weren't randomized on, well, actually, I think this patient probably would benefit from treatment A, so I'll give it to them. Or actually, this patient's got these characteristics, will benefit from treatment B, so I'll give them treatment B. So in this case, then, having that concealed randomization would mean that it's just a chance occurrence which treatment they get, and the investigator or the treating clinician has no influence on the treatment that patient would get. So why is it still important then because of that? Well, it's not the sort of the X Factor band and program on the television, but more crucially, the reason why randomization is important is that actually, if it helps, basically the advantages are if there's an imbalance in known or unknown factors, it would usually allow for that, or will mean that they'll be balanced, particularly if you've got a trial with a reasonably large number of participants. So the crucial thing is that randomization one ensures that there's a balance in both known and unknown factors. And even if there isn't a balance, well, that balance is probably just purely due to chance, and you've just been unlucky if you've got sort of an imbalance. And crucially, sort of in a technical way, statements relating to the chance of getting the observed effects are basically completely free from any confounding effect. So there's a sort of a statistical reason for doing that. So ideally, if you're trying to evaluate a new treatment, then if you can, they should be randomized if it's practical, ethical, and appropriate. So I say practical, ethical, and appropriate, but of course, in reality, there'll be reasons that it's not either practical or even ethical or morally right or appropriate to randomize people. So you have to look at alternative designs. For example, you certainly can't randomize people to sort of smoke or not, for example. So that's why you probably can't have a randomized trial comparing smokers with non-smokers, for example, because it'd be unethical to randomize sort of smokers or not. So that's the reason why you have to look at alternative designs. And as I've said, the main point of randomization is that in the long run, it will produce study groups that are comparable in known or unknown factors that are likely to influence the outcome apart from the actual treatment being given itself. And if there are imbalances, well, you can be fairly certain that they're due to chance. And equally, as I've said, you also need to have the idea also of concealing the randomization sequence in advance, so you can't prevent people from guessing it or subjugating that. So just purely having randomization on its own isn't adequate, but if you can conceal it as well, and that's usually done nowadays with having sort of remote randomization or using the internet to help randomize participants, then we've got adequate concealment. So that's the sort of reasons for randomization. And typically, hopefully, as you can see on this slide, this is typically the standard randomized controlled trial. If you look at this, we decide whether subjects are eligible. We either screen them or check whether subjects are eligible for either treatment. We then crucially then seek informed consent to enter the trial, and patients are sort of could possibly receive either treatment. If the subject accepts this, they effectively go down the left-hand side and then they're randomized. They either get the new treatment or the control treatment, and then we compare the outcomes. Now, of course, if a subject doesn't consent to take part, they refuse, well, actually, they go down the right-hand side and they just get the usual care, control treatment, and then they wouldn't be part of the randomized controlled trial. So that is the very sort of essence of randomized controlled trials. And the problem with this sort of thing is, well, actually, randomized controlled trials tend to typically have what's called good internal validity, in terms of, well, we know that we can make a sort of unbiased comparison between treatment A and treatment B and see whether that intervention works and make some causal inference on that. On the other hand, if subjects refuse, even though they're probably eligible for that treatment, then there may be slightly poorer sort of what we call external validity, because it may be that it's all the young men refuse to take part and all the people who choose to take part in the trial are older participants, so therefore our trial may not reflect, our trial population may not completely reflect the population of all subjects who are potentially eligible for the treatment. But unfortunately, that's the nature of randomized controlled trials, and the only thing we can do about that is really look at when we're trying to assess the quality of randomized controlled trials and the conclusions from them, is look at what I call the baseline characteristics of the participants that were randomized and see how that fits with the either population or patients that you wish to treat or patients you'd ideally like to target with the intervention. So that's the usual sort of RCT design. Well, there are a number of sort of different ways of randomizing participants to trials. The 3 or 4 main ways are sort of simple randomization, blocked and stratified randomization, and a sort of a technique that is called minimization. And some of the purists say minimization isn't really proper randomization, but I won't really go into that debate. But if you're really interested in this, that's this sort of URL for a series of short YouTube videos created actually by a colleague here in ScHARR, and it's available on YouTube by Mike Campbell, Professor Mike Campbell, and how you can demonstrate randomization with some coloured beads and a pack of cards and a couple of sticky labels. So Mike goes in detail and talks about simple randomization, and stratified randomization, as well as what's called minimization as well. If you're really interested in that, there's some short videos available on YouTube that you can actually look at and check out. So that's the sort of the A for allocate at random for randomized controlled trials. The next one, B for randomized controlled trials, is B is for blind assessment of randomized controlled trials. So in this case, we might have sort of double-blind trial potentially. Neither the patient nor the doctor, nor Junior Bear nor Dr. T Bear know actually what treatment the patient's being given. Or actually, you may even have sometimes sort of triple blind, where actually the analyst or the person evaluating the outcomes or the statistician doesn't actually know what treatment the participants have received. So why do we want to sort of have blind assessment of outcome? Well, the idea is really that if you can, it helps prevent sort of bias again. These are the ideals again. Ideally, any assessment of the patient should be blind to the treatment the patient should have received, and this helps you to give basically a more sort of objective view of the efficacy. I mean, but clearly sometimes you cannot blind patients to whether they've received a drug or bed rest, for example, as in the the MRC streptomycin trial. Sometimes it's clearly not possible to do that. But if you're particularly doing the trials in a pharmaceutical treatment, then it possibly is able to sort of package the two drugs or two treatments in a fairly standard way, so you don't know which the patient themselves doesn't know which treatments is received, and even the treating clinician may not know which treatment the patient is receiving. And we've seen this, and I'll give some examples later on in the talk in the with the some COVID trials equally, where you can blind people to which injection they're receiving. Are they receiving an active dose of the vaccine or a placebo dose or an inactive version of the vaccine? So the ideal situation is to basically have the maximum degree of blindness, but clearly that's not necessarily possible or even desirable in circumstances. So it's all possible. Try and have, if you can't blind the patients, if you blind the recruiters, you can possibly blind the people who are assessing outcomes, or even the statistician who's analysing the data could be potentially blind to what the two treatment groups are and just analyse data with codes as A and B, 1 and 2 for the treatment groups. Sometimes that's more difficult. Sometimes you can usually guess as an analyst which treatment they've probably had, but it's not impossible. Ideally, use the maximum degree of blindness possible. So the C for randomized controlled trials is basically got the need for a control or a comparator group. And it's important, if you can, really, for various reasons, to have what I call a control or comparator group to which to compare things. And equally, I think it's probably important to have that control group being what we call a contemporaneous comparison group. And I suppose a classic example now would be that we've got the idea, of course, if we were evaluating new treatments now, we're in the middle of a global pandemic and the impact on health services and things like that. Clearly, the type of patients that's being received, things, and if your control group was historical data 2 or 3 years ago, there may be different types of patients that are being treated and things like that. So that's why it's really, really good to have a contemporaneous control group. Also, things may have changed, because once a therapy, even an existing therapy, is in general use, we don't tend to monitor it to the same rigorous standards as it was during its development. There's probably been changes to doctors or attitudes to treatment and the characteristics of patients who actually get the treatment. These might influence patient outcomes, and that's why it's really, because of these changes and because of sort of external factors as well, that the new therapy is tested concurrently alongside the old therapy. And that's why it's really important to have a contemporaneous control group in the ABC of RCTs. Now, because clinical trials can cost a huge amount of money, then it's really sensible, if you can, to try and design them in such a way that obviously they produce a useful result and can answer a clinical question. And it's becoming increasingly more common now to carry out what we call feasibility or pilot studies prior to the main trial. Now, the problem is with definitions of pilot trials and feasibility studies that we haven't really agreed on a sort of definition of terminology, and even the research funders in the UK have different terms to count for sort of feasibility or pilot studies. But in this case, you can clearly see probably a spectrum of feasibility trials ranging from sort of preclinical on 1 or 2 participants right the way through to the other end of the distribution or the spectrum, perhaps what I call pilot trials, which are randomized and are basically the main trial in miniature, and you really want to see whether actually it's feasible to conduct the main trial. Can patients willing to take part? Are they willing to be randomized? Are they willing to have the treatment and be followed up? And that's the case sometimes where you can have what we call a feasibility trial or a randomized pilot trial. Now, a randomized pilot trial basically could be what we call external, it's standalone, or it could be what we call an internal pilot trial. And I'll give examples of both, but external pilot trials are usually carried out when the investigators expect the design features maybe to change, and so you can't really use the results from the pilot in the main trial. On the other hand, internal pilot trials occur where the major design features are fixed. There may be uncertainty in terms of how many centres are going to contribute patients, whether it's actually possible to randomise and recruit participants, or even the variability of the outcome variable. And in this case, you might conduct an internal pilot trial. Now, most publicly funded trials in the UK now, particularly in my time when I sat on the health technology assessment panel for deciding funding for trials, that we tended to insist for our main trials that they all had an internal pilot phase to sort of recruitment and sometimes retention, and whether actually you could actually recruit to the target sample size. And in that case, the internal pilots usually have to have a point at which you make some decisions about whether to stop the trial or continue, so a stop-go criteria or a go-no-go trial. And a take-home point to remember as people make that the main point about sort of external pilot trials is they're not designed to test treatment efficacy, because they usually don't have a full power calculation, and they're usually too small to be able to detect a sensible effect. And you should really calculate confidence intervals for any estimates and not do any significance testing. But you'll see examples of people who started off, still even published in the literature, intending to go for what they, at the start of the study was a main definitive trial, and because they couldn't recruit to their target sample size, they decide to label it as a pilot trial. And I think such practices are best discouraged. You either start off the pilot trial or you start off as a main trial. I think I've seen one example recently of the other way around, where people started off intending to do a pilot trial but realized actually we could go on and recruit far faster than we wished, and it became a main trial. But such examples are quite rare. So I'll give some quick examples then of this was an external pilot trial I was involved with. It was looking at treatments for COPD. It was randomizing patients to either get what we call early pulmonary rehabilitation or usual care. We had a series of what we thought our feasibility outcomes were, which were basically recruiting 76 participants at 2 centres, collecting outcome data. And the bottom line is we basically only recruited 58 participants. We only managed to collect outcome data on 21 participants. We found it difficult to deliver the intervention, so we basically concluded that a large-scale main RCT using the protocol that we got wasn't feasible. Another example of a trial that's just reporting at the moment was a trial of what we call endometrial trauma in women undergoing fertilization, IVF. And in this case, for the main trial, we were hoping to get 1,044 women from 16 fertility units, and consisted of 2 phases. We had an internal pilot trial to assess the feasibility of recruitment, and then we were hoping to have what we call a 2-year main recruitment phase. And the trial commenced using the same procedures for the main trial, but after 9 months of recruitment, we wanted to see whether recruitment was going okay and where people were actually managing to get the interventional procedure, this endometrial scratch, to before prior to having IVF. So in this case, then, in this example, we actually decided we had some predefined stopping criteria, and we said, well, if we couldn't get 108 participants, stop the trial, and if fewer than 75% of women actually received their randomized treatment or the intervention we were interested in, the endometrial scratch, we'd stop. And as we demonstrated about 9 months, we got more than our target. We got 231 participants recruited and randomized, and of those, half were randomized approximately to get the endometrial scratch procedure, and of those, 85% had actually got the procedure at the correct time point. So we made the decision to carry on with the trial, to carry on recruiting, and then it became the main trial. And actually the main trial did manage to continue and recruited to its target sample size of 1,044 women. And we're just in a reporting stage of that trial now. So that's an example of what we call an internal pilot trial, where we actually met our targets and carried on. Now, of course, substantial amounts of public funds are invested in health research worldwide, and as I've alluded to, publicly funded trials often recruit participants at a slower than anticipated rate, and many trials sadly still fail to reach their planned sample size within the original trial timescale and funding envelope. And I did a review a few years ago of 151 randomized controlled trials published in the NIHR HA journal between 2004 and 2016. I found that just over half, 56%, recruited to their final recruitment target, and it was about 79% of those trials managed to recruit to sort of short of their main recruitment target, but up to 80%. And trials were still asking for extensions. I think in just under a third of them, the original sample size target was revised, and in most cases it was revised downwards. And in terms of recruitment, most trials only tend to recruit at the rate of around about 1 patient or participant per centre per month. But they do tend to sort of on average retain patients, which is good news. And it turns out that around about 89% have valid primary outcome data for you to analyse, which is good news. But this leads to the idea really in randomized controlled trials about the idea of, well, loss to follow-up and what we refer to as sort of protocol violations. So in many trials, some participants will not have followed the protocol, either on purpose or by mistake. Sometimes it will be they've been taken to the wrong treatment, or they've had received the wrong treatment, one that they were allocated to. Sometimes people in reality won't actually take the treatment, call these non-compliers. And sometimes maybe the trial began, well, actually, that patient actually wasn't really eligible and shouldn't have been included. And there's lots of different ways of dealing with these things, but my personal view is that the only safest way to deal with all of these circumstances is, if you can, is to keep all randomized patients in the trial and to analyze as you randomize. So try and remember that. Another acronym, analyze as you randomize, is the ideal thing to do. And there's the two analysts. Maybe I had to guess who those two characters are. So if you choose to analyze as you randomize, then in this case, this leads to the idea of what we call an intention to treat analysis. And it should be based on the groups as randomized. Now, sometimes you may actually perform an addition analysis of only those patients who adhere to the protocol and are sort of compliers with their treatment. And sometimes we talk about that being a per protocol analysis. But for most randomized controlled trials, particularly randomized controlled trials where you're trying to assess the superiority of a new treatment compared to a controlled treatment, analysis groups as randomized should be the main analysis for the trial. Now, occasionally I'm pulled up by referees or people that don't understand randomized controlled trials and talk about an intention to treat analysis. Well, actually, most trials that report, unless there's 100% retention of all randomized participants and that you have valid primary outcome data for all randomized participants, or you make assumptions about any missing primary outcome data, or you impute any missing primary outcome data, then most statistical analyses of RCTs are modified intention to treat analyses. So the estimand that you get for the treatment effect is derived from basically those randomized participants with a valid primary outcome data. And so, strictly speaking, the sample of randomized participants may not be the same as those who you analyze ultimately. So that's the a modified intention to treat analysis, as I call it. Another thing that frequently in health services research, we talk about explanatory and pragmatic trials. Explanatory studies are basically studies that usually seek to evaluate the comparative efficacy of a service and help explain why the service is effective or not. And it's normally, does it work in principle? And usually explanatories usually have things like sort of placebos as the control or comparator drug, whereas in reality, a more pragmatic trial, most people would not be treated with a placebo treatment or placebo tablet or pill. Then we'd be interested in what happens in real life, warts and all. And in that case, we're interested in what we call the effectiveness of a service or technology. And pragmatic studies should aid decisions about which services should be provided. So that's more, does it work in real life? So trying to be topical then, just to prove that trials do this, lots of obviously trials are reporting and comparing vaccines for COVID-19, and I think this was the Oxford AstraZeneca trial, and this trial basically covered the ABCs of clinical trials. It had random allocation. In this case, if you read the text, this time it was still prepared in advance by a study statistician, similar to how Professor Austin Bradford Hill did with the MRC streptomycin trial, but this time it's uploaded to a secure web platform, and the list is held by pharmacists who they say are unmasked, but the staff who are delivering the vaccine were sort of masked to the group allocation. It was actually sort of single blind because basically participants didn't know whether they were receiving the active vaccine or a control vaccine. And even the staff administering the vaccine, they prepared them out of sight of the participants, and people couldn't tell whether patients couldn't tell whether they were getting the active vaccine or a control vaccine. And it did have a contemporaneous control. In this case, it was treatments, a number of different types of vaccines, or even a saline treatment. Now, briefly, I'll talk about analysis. If the trial's well designed, then the analysis can be fairly simple. Then in this case, for vaccine efficacy, we look at something like the vaccine efficacy is 100 times 1 minus the relative risk of COVID-19 in the vaccine group compared to the control group. So in this case then, if you look at the data, I did a rather crude analysis of the COVID vaccine data, and I came up with that the vaccine efficacy based on the data was around about 70%, with a confidence interval ranging from 55 to 80%. And it turns out the more sophisticated analysis that the investigators did, because they had access to all the data and how often people were followed up, that gave an estimate of close to 70% and a confidence interval of 55% to 81%. So you can basically have sort of a simple analysis, if the trial is designed well, is fairly adequate. I'm conscious of the time. I won't sort of talk too much then about superiority and equivalence trials and how to do that, and sort of the other alternative designs. I suppose certain designs, I suppose you can actually, in this case, sometimes you can't randomize individual groups of individual subjects or patients. Sometimes you may decide where you've actually got to do what we call a cluster randomized control trial, and randomized clusters of patients. And a cluster could be a household, a hospital, a doctor's surgery, or even it could be an area or a community. And that's I give a plug for a book what I wrote with Mike Campbell, which is How to Analyze and Report Cluster Randomized Controlled Trials. So that's an example of that. So crucially, RCTs, it's important, and this is basically almost an industry for reporting randomized controlled trials, and it's called the consort statement. And most leading medical journals tend to endorse, that's the crucial word, but they don't actually enforce the consort recommendations. And now there's actually sort of an industry now for reporting of trials now, and there's a website with the consort reporting of trials, and equally there's lots of extensions to the consort trials, and researchers in ScHARR have been involved in adapted designs and also the reporting for pilot and feasibility trials. So that's really some extension to the consort statement that you can get involved with. I suspect we'll get even more and more extensions to the consort statements for the different types of trials. So hopefully this talk's emphasised the importance of randomised controlled trials. I've briefly discussed the different methods. I've talked about the ABC, allocation at random, blindness and control, the idea of having a protocol. I haven't had time to discuss different types of trials except for the cluster trial, and I've unfortunately run out of time to distinguish between superiority and non-inferiority trials. But you've got the consort guidelines as well for reporting and the trial results. So if you're really interested in these things, I'll give another look for a book. You've just missed out. Christmas is just gone, but here's a book that I wrote with Mike Campbell that talks about some of these issues in more detail. So thank you very much for listening. Any questions?
Okay, thanks, Stephen. Yeah, if anybody wants to post any questions for Stephen in the chat box, feel free, and he'll do his best to answer them. And you should see further up in the chat thread, Andy has posted a link to the next session with Dr. Liz Crute. So if you want to book for that, we are currently arranging the rest of the program for 2021. I had to think then for a minute about what year we're in. Yeah, we're putting together the rest of the program for 2021, but there's going to be, there'll be a session, a mini masterclass every month in 2021. I can at least reassure you of that. So you should be able to book for some of the ones beyond February very soon. But in the meantime, you can book for Dr. Liz Crute. Oh, sorry, I can see that Andy's just posted to say Hannah Jordan is going to be talking in March. Hannah is the program lead for our Masters in Public Health. So yeah, so she'll be talking. Do you know what her topic is, Andy? Or is that yet to be decided? To be confirmed. We will wait and see. I can tell you that Liz is taught. Well, I think I mentioned it at the beginning, didn't I? But Liz is talking about doing research with people with learning disabilities, I think. Emma, I can see you just raised your hand. Do you want to post something in the chat box?
I was just trying to see, actually. I was going on my chat, and is one of my settings, I'm only seeing the moderator's chat. So unless anyone's actually posted questions.
Okay, I can see Nicola had her hand raised. Do you want to ask a question, Nicola? Sorry, I'm just zipping back and forth through my various chat threads. I could see that Nicola was raising her hand.
I was going to say yes, so it may be because I can just see the moderator's chat, if that makes sense. And I'm thinking, am I missing something here?
I can see everyone. Okay, in that case, I think, I'm not sure how the settings for the session have been set up. I'm really sorry about that. I would imagine if you can't see the chat, then it's possible that the session has been set up so that you can't use your microphones either. But I'm not entirely sure.
Okay, I just had a quick check. It was set up with the 250 kind of participants limit, which disables the chat. I've switched that off, so the chat should work now. So sorry about that.
Okay. Luke, will people be able to see that immediately, or would they have to log out and log in again?
It should be on. Let me just click save again in case it didn't take it the first time. Try now.
Okay, can anyone now see the